Hi, my name is Philip Baker. I'm a senior biochemistry major, and I was working with Dr. Vidisha Singupta on performing a bioinformatic analysis on biofilm formed by Bacillus thuringiensis in wastewater treatment plants across East Texas. A biofilm is when a normally single-cell organism comes together to form a multicellular organism, and they attach to surfaces and each other by secreting what's called extracellular substances. And in this project, we grew our biofilms in wastewater treatment samples from San Agustin Wastewater Treatment Plant, Nacogdoches Wastewater Treatment Plant, sterilized water, and tap water. To study these biofilms, we first used scanning electron microscopy to visualize and measure the differences in structure and thickness of these biofilms. And we saw that the biofilms grown in the Nacogdoches wastewater treatment sample supported a much greater growth of biofilm than compared to San Agustin wastewater treatment plants samples and sterilized water and tap water samples. And that led us to believe that there may have been a genetic component that was causing them to grow significantly more. To test this, we sent our samples to a company called Novogene, which is a biotechnology company for RNA sequencing and transcriptome analysis. And from the data that they sent back to us, we saw that the chemical composition of wastewater can induce genomic changes within biofilm. We saw differential gene expression, which is when genes are expressed either higher or lower depending on their conditions. Specifically in our Nacogdoches sample, we saw that there was significant enrichment in genes related to ribosome biogenesis and protein synthesis. And both of those are essential in the construction of the extracellular polymeric substances, which acts as the glue for the biofilm to stick to each other and to attach to surfaces. In our San Agustin samples, we saw that there is significant enrichment in genes related to nutrient uptake and nutrient distribution, which is crucial for sustaining a large multicellular organism such as biofilm. Overall, we saw that all of our biofilm samples showed significant enrichment in genes related to the formation, growth, and sustainability of biofilm, and that the chemical composition of the wastewater strongly influences the bacterial's gene expression. And I would like to sincerely thank the S.U.R.E program for allowing me to do this research.